Welcome back, everyone, to DrAariaWorld Science School, where we learn advanced science concepts in a fun way and apply them to solve very important problems. Today is very special because we're approaching the end of the year and also my birthday. So today we're going to recap some of the topics that we've covered this past year and also offer a sneak peek of what's to come. Last year, we launched our science school with a focus on chemistry and its applications. We started with a review of the atom, the periodic table, and then after that, we moved on to different types of chemical bonding, such as covalent bonding and ionic bonding. Since that period of time, we have moved on to more advanced topics in chemistry. This included molecular modeling. We did this in different ways, and we showed that three-dimensional representations of molecular models are much more accurate and provide more detail than two-dimensional representations. We also showed ourselves some 3D representations using molecular modeling sets and kits. Then after that, we moved on to some more advanced types of bonding, such as coordinated or dative bonds. Remember this? We created molecular models of some of the most important chemical reactions in photosynthesis and indeed in biochemistry. I do hope that you remember this. This is our molecular modeling board, showcasing some of the steps in the Calvin cycle, among other things. Then we discussed biological macromolecules, such as proteins and DNA. We also talked about certain proteins called enzymes to help speed up chemical reactions, including, for example, those involved in photosynthesis. We then discussed how one such enzyme called rubisco controls the rate at which carbon dioxide is captured from the atmosphere by plants and certain marine organisms. And we're also starting a project to make this enzyme more efficient in the lab to curtail greenhouse gases and fight climate change. I'm just going to show a small video. So as you can see, these little blue lines is rubisco, and here in the middle is ribulose 1,5-bisphosphate, a crucial molecule in the process of the Calvin cycle. And over there is a little carbon dioxide. We also looked at this video and rubisco, the enzyme, from different magnifications. For example, like this. This is rubisco from a little bit further away, and now we're zooming closer a little bit more. Then we talked a little bit about spectroscopy. And here is ribulose 1,5-bisphosphate we're taking a closer look at it. And here are the individual atoms. And when you press over here, like this, you can see the distance between two atoms and the distance of the bond. So here it says 0.153 nanometers. Okay, this is a very interesting experience. Now we're going to talk about our sneak peek on different steps. We're going to be studying chemical reactions and a subject called stoichiometry. This allows us to write chemical reactions and balance them to calculate how much of one compound is needed to react with another compound and how much is formed from such chemical reactions. This will allow us to calculate in particular how much CO2 can be captured by photosynthetic reactions under certain conditions. And here are we're going to talk briefly about some of the different types of chemical reaction equations, such as rearrangement, combination, and dissociation. For example, A and B react to form C and D, rearrangement. A plus B become C, reacts to form C, combination. And A dissociates to form B and C, which is dissociation. So the first step of balancing a reaction is to first write the chemical formulas for each compound on the left and the right with an arrow between the left and the right. This means that molecules on the left react to form new molecules on the right. The number of each atom cannot change. So adjust the coefficients A to D for compounds that contain elements appearing in only one compound so that the numbers of those atoms on the left/right match. Then adjust the rest of the coefficients A to D to match the numbers of remaining atoms. Now we're going to balance the chemical reaction for combustion. So the first step is to add A, B, C, and D in the equation. A molecules of glucose react with B molecules of oxygen to produce C molecules of carbon and D molecules of water. So step 2. Carbon only appears in glucose and carbon dioxide. So choose A is equal to 1 and C is equal to 6 to match the number of carbons. The third step is to match the number of oxygens and hydrogens on the left/right to find B and D algebraically. 6 plus 2 multiplied by BOs on left, 12 H's on left, 12 plus DOs on right, 2 times DH's on right. Match the hydrogens first since there is only one variable. So that's 12 is equal to 2 times D, or D is equal to 6. Then we match the oxygens by choosing B. 6 plus 2B is equal to 12 plus 6, and hence B is equal to 6. So 1 molecule of glucose reacts with 6 molecules of oxygen to produce 6 molecules of carbon dioxide and 6 molecules of. Thank you so much for joining me here today. I'm really happy that you joined me here on this video, and I hope that you enjoyed and like the material that we're going to be studying in the future. Bye. We also studied DNA in the lab. We started collaborations with multiple organizations to study the effects of climate change on organisms, including endangered species in the oceans. These included so first of all, at home, the Morristown Save the Environment STEM organization, we collaborated with this organization, and we successfully identified what fish species and how many of each live in Morristown streams in different seasons using DNA analysis alone. The National Oceanic and Atmospheric Administration, we also collaborated with this company. It is here as shown. We also collected water samples from the famous Channel Islands for some of the first DNA analysis in this area. And are currently analyzed. We also plan to track endangered species in these islands, called the Galapagos Islands of North America, in a reference to Charles Darwin over time. Jenkinson's Aquarium on the Jersey Shore, where we proved that the accuracy of our DNA-based species identification by successfully identifying over 95% of the fish species that there were, examples, including endangered species such as the sand tiger shark, which we now intend to track in the ocean. We also showed how one can set up a home lab to extract DNA by filtration, get it sequenced, and use and analyze the data in one. Here's a video about how we put the alodia leaf, which is a pond plant, under the microscope, and these are how the chloroplast are moving. So as you can see, the chloroplasts are actively moving because this was still a fresh sample. And when the samples are still fresh and have recently taken out of the water, the chloroplasts move quickly. And as you can see, they're actively moving. But yeah, it's very cool and interesting to look under the microscope like this. And because then you get to see the chloroplast moving underneath your own eye. And also this last picture, it shows how we are performing the process of environmental DNA filtration. Thank you so much for joining me here today for our science recap and steps that are to come. I really appreciate you joining us today. Thank you.